Today we're going to be talking about the cell cycle. And to get us started, I want to talk about a chromosome and what a chromosome looks like. There are three ways that a chromosome can present itself. One of those ways is in a way called chromatin. And with chromatin, all chromatin is, if you ever hear us talk about it, is it's relaxed DNA. It looks like a string in a small little ball, just in the way that I just drew. Or like spaghetti, just like that right here. Moving on from chromatin, another one would be called chromatid. If something is talking about a chromatid, or if we are talking about a chromatid would just be a normal chromosome, which is just usually represented as an X, and it is split in half. A chromatid is just half of, or one side of, a chromosome. And that's just half of a duplicated chromosome. That's how we like to represent that, just half of that one side. Our next side, or our next type of chromosome, is just a chromosome. It's DNA, and DNA being that it is our big, long trail of, let me change this color, trail of phosphates and sugars, and it's in this shape. It is all of this, and DNA is really long, and all it is a condensed DNA, that long strand of DNA, condensed and put into a big X, just like that. And that's how we represent our normal chromosomes. It can be duplicated, and again, it looks like an X. It can also be unduplicated. And in that case, it would just be that half, again, just one of those chromosomes that's not duplicated. And so as we look to the next part of our notes, we're talking about, in this next lecture or lesson part, we're talking about the cell cycle. And the cell cycle is just the period from the beginning of one cell division to the beginning of the next cell division. So we have cells all over our body. We looked at the cells inside of our cheeks just the other day, and inside of our cheeks, our cells look like that. And they usually have a nucleus, which is in the middle, which is where all of our DNA is. And this cell needs to sometimes replicate itself. And the cell needs to replicate itself just like DNA does. And so a cell cycle has a life cycle represented from the period of the beginning of one cell division, this cell dividing in half, to the beginning of the next cell division. And this picture is what we're going to label here in a second. So, the cell cycle has two main parts. Our first part, and you're going to write this part down, part one is called interphase. Write that down. Interphase is where our cell grows and prepares itself to be divided. Grows and it prepares itself to be divided. In part two, again, you're going to have to write down cell division for part two, and it's got two parts to it. One of those is called mitosis, and that's where it's called nuclear division. And then the next part is called cytokinesis, and that's where our cytoplasm divides. Nuclear division is talking about the nucleus. Nucleus. And that's where the nucleus divides. That's where our DNA gets split in half. And that is, again, mitosis. So two parts to our cell cycle, that first part being interphase, and our cell is just sitting there, it's growing, it's preparing itself. And then that second part is when the cell finally divides or splits in half. So, interphase. In this stage, the part that you're going to write down is that the cell is engaged in regular processes and it prepares itself for mitosis. So, for example, a cell that is on our muscle is going to be performing its regular process. Look at that, I can't spell muscle. Is going to be performing its regular process of working for us, lifting things or helping us walk. It's performing its muscle duties. It is also probably going to be doing or preparing itself for mitosis. So it might be getting more DNA or doing things like that. A cell that's on our skin, A skin cell might be just being there, blocking everything from coming inside of our body, like diseases. So it's performing its normal duty or normal function. And so moving along with interphase, interphase has 3 stages. It has a stage called G1, S, and G2 stage. And so here's what some of those look like. G1, S, and G2. Interphase is the longest part in the cell cycle. Whenever we look at cells, which these are a whole bunch of cells inside of an onion, you can tell that we have a cell sitting right here, and on the inside of it, we have a nucleus, or what we call a nucleolus. I'll get to that in a second. But most of the cells sit in that nucleus stage, in that interphase stage. Interphase is like growing up. So your parents usually develop you into a mature adult, and then there's a cell division, which is mitosis, and you can split from home. That analogy of you splitting from home and then beginning life on your own. So here's what that could look like. Interphase is divided into 3 stages. In GAP1 phase, your cell increases its size. It has a checkpoint, and it makes sure that DNA is ready to be synthesized. Synthesize means to make. To make. So that's what you're going to be writing down as your G1 phase. Your cell gets increased, and then it hits a checkpoint. What I like to think of that checkpoint as is a stoplight. Is a stoplight. Just checks it to make sure that it's ready to move on. Once it's ready to move on, it goes through that checkpoint. It moves on to the next phase, which is called the synthesis phase, or S stage. In this phase, DNA gets duplicated. This is where that DNA replication comes in. That process that we did through replication using enzyme helicase and polymerase, all of that is happening inside the cell during S phase of interphase. I represent that as an I. Our next one is called G2 or GAP2, and this is where the cell grows. It produces some proteins, but it mainly grows, and this is our second checkpoint where we make sure that DNA was synthesized correctly, so that we make sure that the order of A, T, G, and C is correctly replicated to a T, A, C, and G. And if there's any mistakes here, maybe we accidentally put an A here, that this checkpoint would be able to say, this is wrong, let's correct that. All right. And so you can represent this as that analogy that I said before. You, in G1, your parents begin developing you into a mature adult. As a baby, you grow and you go through kindergarten and you get all these good grades. You make sure that you're ready for the next phase in life, which is you should be ready to move on. And you can go into high school where your S phase is, where you get the correct DNA, you get the correct grades. Now you're ready to move on to your last one, which would be like college. And this is another teacher here in this school here. Oops, let's go back here. Okay, so it checks itself and moves on. So what interphase look like? Interphase looks like a cell. So this is our normal cell. DNA is in chromatin form, so you'll see a black nucleus down here in the middle of the cell, and usually your DNA is in chromatin forms, or that string form. It's in string form. A couple of things that are going on here is that your chromosomes are not clearly visible yet. So they're just going to be a really dark spot in the middle. If you see a cell that has a dark spot in the middle, it is probably in interphase. There's these things called centrioles, and they're present in animal cells. That's important to remember. They're only present in animal cells, and they're going to be used here in a little bit. So, next, after interphase, the cell goes through cell division.